stochastic evolutionary control of therapeutic resistance in heterogeneous, like cancer or microbiome population. So our goal is to reduce the population mean fitness by optimizing the drug sequence. So it's also because of the colloidal sensitivity, because some different genotypes respond differently to each drug. One genotype might be resistant to drug A, but sensitive to drug B, while another one can behave opposite way. So the optimal drug depends on the current genotype frequencies. So we first model the heterogeneous population using right feature dynamic with multiple genotype. And each generation, the genotype frequency fluctuates due to the mutation, drift, and the drug dependent fitness. And we allow the drug to change each generation. And for large population, this dynamic can be approximated by the Fokker-Plot equation, which describe how the probability distribution of the genotype frequencies evolve over time. To make this checkable for the MDP, we project the 3 genotype simplex onto a square, and then finally decretize it into a grid. And from this, we can derive the transition probability matrix. And this grid become the space of the MDP. And the action will be the drug, and the reward is the negative fitness. So maximizing the reward corresponding to minimizing the population fitness. So at the bottom left, panel C and D shows a toy example of how the population moved under the Shepherd protocol from generation 1 to 8, and different colored region indicate which drug is optimal at each genotype state. And on the right, we apply the MDP derived drug policy back to the right feature simulation, and then we compare the Shepherd against like single drug treatment and 2 drug cycle. And I only show the result for 3 genotype cases here, but we also test it in higher dimensional system, including 4 and 8 genotype, and Shepherd consistently maintain lower mean fitness than any other protocols.